Well, thank you for the nice introduction, Nick, and the invitation to be here. I have to say it's been a very nice 2 days here in Cambridge, interacting with people from various departments. And so it's great to see that this multidisciplinary aspect of thinking about origin and prevalence of life is expanding to many different universities around the world. So today I want to talk about water-rock reactions in the search for life. And I heard that actually this may be a recurrent theme, as I heard that there was a speaker, Barbara Lawler, one of my collaborators as well, that was here and actually told you a bit about some of these water-rock reactions in hydrogen, probably in the subsurface. So there'll be a little bit of that, but this is good because then we have had a background on kind of what is there, and then we can have a little bit look forward into what this means for life and evolution and microbial diversity on Earth, and a little perspective of what that might mean in the search for life elsewhere as well. So to begin, I'll start with just this theme, which some people are often surprised to hear, but actually is what we have observed by looking at the subsurface and cellular abundances, is that actually the majority of bacteria and archaea live underground. So these are from the 100-meter depth to where the temperature limit of life exists. And what's surprising is that this is a very large biosphere as far as quantity of numbers of cells, but it's not just purely the amount of cells that are living actually beneath our feet, but actually these large number of cells, there's actually an incredible amount of microbial diversity that also exists. So as many of you know, there's been sort of this genomics revolution that happened about the last 10 years, where more and more DNA sequencing has been done. We've been looking at microbial diversity, and this here is just a picture of the archaeal tree of life in different habitats in which microorganisms have been found. And what you can see is that there's this red, the sort of environments that we're looking at in here, such as the vents, the acid mine drainage, the subsurface, these colors of green, purple, and orange are showing up, having representation all the way across the archaeal tree of life. And in fact, many of these new branches of microbial diversity that we're seeing through metagenomic sequencing, these groups that we call like the loci archaeota, which are potentially this bridge to eukaryotes, look to be a bridge to eukaryotes as far as archaeal diversity goes, this massive superphylum called the Potassiobacteria, the candidate phyloradiation within bacteria. These are very strongly and heavily represented in the subsurface. So many of these very divergent, deeply branching groups that don't look like our typical tree of life that we, at least me, myself, maybe you undergrads are now getting kind of this revised tree of life with the genomics revolution, but this traditional tree of life has tremendously expanded, and a lot of that has to do with increased diversity that's been seen in subsurface environments themselves. Now, there are basically two reasons why we can say that, okay, it makes sense that the subsurface is holding such a diversity of lineages. And that's one, of course, because if we just think about this larger population of things, we have more species representing based on some sort of scaling type estimates. But another reason, more reality as to why so much diversity is sitting down there, is because of the variety of water rock reactions that can exist, the variety of mineral phase, the variety of oxidation states that are existing within the subsurface that can be accessed. And so various types of metabolic networks emerge, various centrifuges appear, and they are spread out across our continent, our planet's subsurface. Now, as we are thinking about not just diversity of life on Earth, but sort of this perspective of what life may look like on other planets, you can think that not just take just any subsurface environment, which of course has interactions with our surface biosphere, but we can go down to sort of this end member subsurface ecosystem, and what does that look like? So what does microbial life that lives underground look like when you remove all of the surface processes and are looking at ecosystems that are purely sustained by the interaction between water and our planetary surface, or these water rock reactions? And so this kind of environment is actually accessed by going into subsurface environments and looking into deep groundwaters that have no detections of that have extended resonance times and very little organic carbon or oxygen, dissolved oxygen, etc., in the waters themselves. And what we look at when we say this sort of extreme subsurface end member is that most of the subsurface life living there is actually supported by hydrogen. And so this is, a very typical reaction that is often talked about in sort of origin settings, in thinking also about subsurface life itself, is a lot of attention is given onto serpentinization. And so here is one type of water rock reaction, a hydration reaction, that can produce large amounts of hydrogen, which microorganisms like methanogens can take and fix carbon and actually sustain various ecosystems. And they occur basically by the oxidation of some ultramafic rock like olivine and end up becoming serpentine, reduce the water into hydrogen itself. And of course, there's on Earth today, there's a variety, I think there's some people who study this quite well, a variety of lithologies that have that undergo serpentinization, producing hydrogen in the environment. And of course, depending on what exactly your geological context is, whether you're, say, a slow spreading region, the mid-ocean ridge, or maybe something at a continental margin, you'll have different rates of hydrogen production in these environments. Another hydrogen generating process that is very important for subsurface environments, which is also perhaps less discussed about, is that of radiolysis, which is the decay of elements like potassium, thorium, and uranium that are radionuclides that are decaying over time, and that decay is able to split water. And so here just a nice illustration to kind of think about what goes on during radiolysis made by Quantum Magazine, where you can see that in various environments of the subsurface, like either sediments or the continental crust, we can have the radionuclides decaying, splitting water, which then we have radicals that can form hydrogen peroxide, and also in the case of in the continental subsurface where you have nice minerals, it's been shown that this sort of radiolytic process can actually lead to the oxidation of minerals to form things like nitrate into subsurface waters. So we can...
Metabolism like when it's powered by hydrogen, there's no oxidative phosphorylation, there's nothing of that order. How is it powered?
How is what?
Metabolism.
So metabolism, right? You can have substrate level phosphorylation happening, right, by the processes of these organisms, and they're harnessing basically this redox potential through the coupling of things like using hydrogen and CO2. So this is the way to gain energy off of this process. So by having this reductant, now they can use that in a redox couple, to have energy to power biological processes. So that's this big question of how do you actually get. So the reason why we're talking about hydrogen is that we want to power by hydrogen because we want to have this reductant in the environment, and we also need to have something, right? So, and then we also want to be able to couple this to various chemistries. And what's surprising about this process is we can actually we can actually, so with this is that we can produce, so we can actually take, say, generate hydrogen, and we can also oxidize into sulfate, and we can then say reduce sulfate, oxidize hydrogen, right? This can be a sulfate reduction metabolism, right? And so this are various processes that can geological processes that can provide molecules that can then be used by life to power themselves. And so these reactions are quite, while the important thing I think I wanted to point out is that while serpentinization is of course, you're really wanting these iron-rich ultramafic rocks. One thing about radiolysis that is surprising in what has been found in the literature is that, right, we can think of any sort of rock containing these radionuclides decaying, so you can have more widespread hydrogen production in a wider variety of places across the planet. Now, the truth of the matter is, while many rocks can have these radionuclides decaying, the amount of hydrogen produced is still bigger in serpentinizing environments, even though they're more localized, than with radiolysis itself. But the amount of hydrogen that is produced through these water rock reactions on our planet is actually quite large. So perhaps Barbara showed this picture in this table from one of her previous works, looking at basically the hydrogen production per year in a variety of environments. And what the reason why she's looking at this estimate is because actually in the continental subsurface, certain environments are even extremely enriched in hydrogen. So these, the work of how much hydrogen could be produced and how could hydrogen accumulate over time is something that was looked at to explain why certain environments were holding, in some cases, 80% hydrogen in their dissolved gases. And actually several of these Precambrian waters in Precambrian rocks were having over, say, 30% hydrogen in their water themselves. And this is largely due, of course, in part by radiolysis, which is listed at the top, the estimate for radiolysis. And then, of course, these hydration reactions like serpentinization, providing additional delivery of hydrogen. At the same time, such estimates like this have since been made about how much hydrogen can be generated, because the reason why we're all interested into this hydrogen and how much hydrogen is because hydrogen is the base of these microbial ecosystems, these large microbial ecosystems that are living deep underground. And various estimates of these hydrogen productions in Mars, as well as in Enceladus, have been made. And while they look smaller than what we see on our Earth today, important in this study looking at, I couldn't recover which depth exactly they went to, but they were modeling basically the amount of sulfur sulfate that can be reduced based on this amount of hydrogen and can sustain reasonable populations in the Martian subsurface based on this relatively lower hydrogen production rate just by radiolysis. And on this here, on Enceladus, what's remarkable is this hydrogen production rate that's estimated due to the increased porosity of this crust. This is only the first only the first 2 kilometers of the Enceladus crust, so a much smaller volume, producing quite a bit of hydrogen. So what we can take from these 2 studies looking at hydrogen production that is estimated to happen on other sort of rocky bodies, that this could be potentially enough hydrogen to produce a sufficient redox energy and sustain a habitable environment in the subsurface of present-day Mars. And in fact, a nice thing to also think about in the context of hydrogen and how much hydrogen there is, don't have the clicker, so, is that with radiolysis in particular, you can see that early Mars, the Noaki, right? We have, since we're looking at radionuclides, these are decaying over time. So we have less and less as time goes on. But earlier in the Martian history, right, somewhere around 4 billion years ago, this hydrogen production rate would have been actually additionally an order of magnitude higher than it is today. So this is a very good source of potential energy for potentially life to be able to harness and use for their own metabolism. Okay? And so this is, putting a little bit of the cart before the horse. So midway through, already saying that, what we would like to say is that, okay, there's very interesting phenomena that happens when you have water and you have rock, and specific water-rock processes are sufficient to generate metabolites that are needed to power and sustain life. And that places like either early Mars or even Mars today, if water is existing, there should still be enough energy produced so that life can use it and sustain itself and live just like life lives deep underground today. And this is a picture here now just about this idea that, that potentially at some point, early on, Mars lost some surface water, but right now it's still unclear whether or not the Mars subsurface has an actual groundwater system. Some indications suggest that this groundwater system exists and is actually stored at a depth with reasonable porosity and temperature, in which life, in fact, life that maybe looks very similar to the subsurface life that we see on Earth today, could sustain itself by these water-rock radiolysis-like reactions. So if we actually did bring some sort of life early on in Earth history, we know that chemolithotrophy, which is what these microorganisms who live underground on Earth today use, evolved very early. There could potentially be organisms that emerged, found refuge in the habitable Martian subsurface, even though the surface of Mars appears to be more or less inhospitable. And so this sort of brings me to kind of the second half of what I want to say, is I hope I've convinced you that, subsurface life is an interesting place to look, that, just by having specific reactions of water and rock, we can create things like hydrogen. And of course, I'm sure there's been many other talks about different types of how mineralogies and, metals and things like this can be used by life itself. But I thought I could bring a different perspective into the problem and think about, okay, now say, is there habitable life in the Martian subsurface today? Was there enough time for life to actually evolve to be capable of utilizing this radiolytically generated hydrogen that could potentially be in the Martian subsurface or other planets? How much time would you need, and how evolvable are living systems sustaining themselves off of these water rock reactions? And so here's just a nice picture from Reprieve's PhD advisor, T.C. Onstadt, who's unfortunately passed away, but one of his final papers was kind of this outlook about how long was how long was there surface water on Mars, and when exactly did specific types of metabolisms emerge, and how long might it have taken. So here you can just see a nice picture of sort of the timescales at which major transitions in microbial metabolism happened on Earth relative to when surface water existed on Mars. And so the main takeaway here is that, anoxygenic photosynthesis on Earth, we know it's of maybe somewhere in this 3.5 billion years, but life has been active well before that. And before life emerged, we had these chemolithotrophic organisms and the ability to fix CO2 based on our genetic information that we've recovered. And the fact that we could have chemolithotrophy actually really early and well before photosynthesis, as far as in evolutionary time, so what it appears that it could be several hundreds of million years before we have actual photosynthesis, but that this could be a viable timeline in which, we could be able to utilize this hydrogen. The main thing is, okay, photosynthesis may never have emerged on Mars because that might have taken a bit too much time, but, the precursor that we see, at least on our evolutionary history, this ability just to utilize hydrogen and fix CO2, this could have emerged within the timescale in which the surface of Mars was habitable. And so then this kind of leaves a question, I think, for me, if I'm thinking about, okay, so now we can do a thought experiment. Since we're in the Leverhulme, a fun one is, okay, let's say that a chemolithotrophic system evolved on the surface of Mars, but it died off. The surface became inhospitable and it had to go into the subsurface. How far could have life have evolved from then? How much diversity could have generated? How complex could life itself have gotten? So I think this is kind of a fun puzzle just to think about. And I'll tell you kind of at least what we observe now in the subsurface as far as how far have microbial lineages evolved, what is the basic genetic diversity, and interesting patterns we see in the subsurface. Okay? And so to get to this question about, what could have happened if we had a chemolithotroph sitting on the surface of Mars, and now 4 billion years later, we go and we finally go look and maybe we find life. How far could it have gotten? And so on Earth, if life is on Mars or say another planet is operating in the same way with some sort of genetic code, we know that there's a variety of genetic diversity generating processes that can lead to what we call evolution, right? And so here's various mechanisms on Earth at which variation is produced in genomes. So this is the one that I think everyone is very familiar with. We can have mutations, so we can have single changes in our DNA. Maybe you remember things of meiosis, and you have recombination, crossover of chromosomes, and then of course getting to gene transfer, and actually then movement of. So here are movements of genetic elements, of horizontal gene transfer, plasmic acquisition, and then things like here, which is a specific molecular construct, the CRISPR acquisition, which is actually a specific genetic element that is a sort of defense mechanism or immunity response of microbes against viruses. So there are also, so these are different ways that organisms are changing their genomes, and particularly microbial focused. And we have a question?
Yeah, what makes horizontal gene transfer more deterministic than mutations? Maybe this goes in a direction you don't want to, but
yeah, so usually this is thinking about if a horizontal gene transfer is kept, like there's some sort of cost because it's going to be a larger piece of sequence rather than just say mutation. So mutation might just be a small thing, and it may not even lead to a change in function, right? We have this idea of synonymous, non-synonymous. And so, yeah, this is, thinking of a gene being a functional element. It's a big element, so it will more likely be following and staying in because of the fact that you're leading to some function. and that there's this bigger cost associated with, carrying the gene.
Okay.
Yeah. And so we have basically, these ideas that, of course, we have different types of genetic variation and then also different ways in which they may affect, populations themselves. So some things just maintained, staying and causing variation, but not having too much effect, or certain things that may actually lead to, say, a change in function that may actually give you some sort of fitness advantage or fitness disadvantage and cause something more like selection. So now I'm going to get into a bit of some sort of genomics and looking at sort of how there's differences between organisms. So a little bit of a different approach that maybe some of you have seen before. So this is sort of the trigger warning of what we're going to show is a lot of charts that maybe look like trees and heat maps. And so let me just describe what we're looking at. So here I'm going to show you what basically a species of bacteria looks like when I compare the genomes of many different bacteria. And so to make it a bit easier and thinking about to begin with, I'm first starting with this idea of Prevotella copri, which is a bacteria that lives in the human gut. So the way that you can think about this is that there are people, which are, for privacy reasons, put by letters. So I have patient K, patient BVE, patient AK, T, etc. And each of them have multiple different types of this bacteria, Prevotella copri, living inside of them. And when I take out these bacteria, I can then sequence them and basically place them in a phylogenetic tree to know their relationship. And when I put them here in this phylogenetic tree, I can look at the similarity between the genomes is related by the distance between them on this phylogenetic tree. Things that I can draw out right away is that there seem to be 4 major clades of this specific species. One is very distant, looks very different from everything else. And then there's these 3 groups. So this is also something not by itself. And for those that look at trees, it's not so many, but this has also been this is a subset of clones, but was supplemented with much more diversity. So there's many members that actually lead to these clades. So no need to worry that there's just one. So these are real clades. And we also can see that some people have, many species and some don't have a few. And so that's what you can get out of here is that there's basically different populations of Prevotella copri that have evolved over time. And I put some stamps here which give us dates that we can estimate based on also Prevotella copri that we pulled out of the Icemen. I didn't pull them out, but they were pulled out by some other people. That's in the Icemen and also coprolites that you can find actually genomes of this specific species of Prevotella copri, where we can then recover its genome and also place it in a much larger tree to estimate the divergence age. So we know that these different populations have actually been separated from each other very far back in time, much before these people, these specific set of patients who are sequenced in New York. The next thing I can see when I look at kind of inside the genome is not just these markers of evolutionary time, these phylogenetic markers that lead to my phylogeny and allow me to estimate the divergence time of the different groups. But I can also just see which kind of proteins are present in the different genomes across my various patients on the y-axis. So you can think of like if I was to take the genome of a single species and I was just to pick all of the proteins and put them in a line. And over here are the proteins that are seen in the most genomes. And over here are proteins that are seen in less genomes. I get in blue, of course, the proteins present everywhere, which is what we call a core genome. And then I can see that there's quite a bit of variety here on what I call the flexible genome. And what is interesting when you look at Prevotella is that there's quite a bit of variation between patients, specific blocks of genes that correspond to the individual patients that may not actually correspond to that don't actually correspond to their placement in the tree. So these are genes that are very specific to their host, that are across everybody. But there are also genes, which is a bit harder to see because it's placed by the clustering is by patient, because that's where there's more similarity. There's also still genes that are clade specific. Okay. Okay, so this is one way that we can basically show in a heat map about this kind of genome-wide comparison and get a sense of how similar different organisms are. And I guess one thing I should tell you that what is quite interesting here, just as a side note to the biologist, to not throw out the biology, is that actually what's patient-specific in the populations is that this has to do with the diet, which is quite interesting. So it's what you eat in so specific carbohydrates that the microbe wants to degrade because you have a tendency to have one diet or another. That's actually what's sort of at the population level in the host being selected for because of these, which are on more sort of recombination type interesting loci. And so that's actually very flexible. And that can undergo much faster and is not actually driven by the placement in the evolutionary time. Okay, so then now, so that we don't look at 500,000 heat maps of different genome comparisons, they're going to kind of compress it into something a little more, another way to visualize, where we now can think about that information, where the genome alignment is more or less thinking about that sort of heat map idea. So how many of those proteins are being seen between two organisms? So percent, if you have complete blue on the heat map all the way across, that'd be 100% alignment. And as you start having different variation, your alignment score is going to decrease. Sort of like this picture being something like 90% alignment. And then you have another term, which is metric to compare genomes, which we call the average nucleotide identity, where now we can see that maybe on this chunk here, we saw that there's one mutation. And so now we have, I think these were 90% genome average nucleotide identity. So that heat map, if I was to project this information about Prevotella copri onto a heat map, which is something, this is some idea by these guys, we can see that a metric that was introduced first by Ohm et al. is that we can see Prevotella copri, the genetic diversity. These heat maps, we had something like 75% genome alignment, so that flexible genome was out there causing a decrease in the y-axis. And if I compare all the parts between them, most of the organisms have high nucleotide identity, but we have a sort of distance block here, largely due to these two members being rather distant to everything else. Okay? And so there was a very, as Ohm did, which is a very nice study, was to start looking at these sort of species boundaries across many different environments by taking this idea of, okay, now let's not just look at one species, but let's look at everything that's living in a single environment, compare the genome to see, within that, how diverse are the species themselves. What was interesting to see when this analysis is done is that in the ocean, we have very high, we have this very hot area of rather high ANI and less flexible genome, but it's still sort of more smooth across the species boundary than some aspect of a soil plane where you have much less geographic distance. So this is just several kilometers, tens of kilometers area in California versus the Global Ocean Survey. There's very discrete patchiness and not this sort of continuum in the diversity distribution itself. And so then, as I said, we're kind of thinking about the subsurface. So what actually does it look like when you look at the subsurface and the organisms that exist there? So is it going to look more like the ocean or more like the soil? Is it going to look more like the ocean here, well mixed, or more like the soil, or is it going to look like something else? And so what I'll show now is data that has come from Census of Deep Life, which is a big collaboration run by the Deep Carbon Observatory, where a variety of subsurface locations from around the world were sampled and analyzed to assess the microbial diversity. Within this data set, what we call MAGs, or these metagenome assembled genomes. There are over 200 of them found in 2 or more locations. And so then we can provide these pairwise comparisons of all the genomes to see what they look like as far as the diversity. And so what was quite surprising when you look at the soil to subsurface or ocean to subsurface is that subsurface is very much a sharp tendency to come to the very top, to the top right. So this was kind of interesting to see that almost everything has a very high nucleotide identity and a high genome alignment. But the caveat here in this analysis is that if we look at the map here, the sampling scheme was designed to be a global survey to maximize the diversity of Earth. And so not too many samples were taken within a relatively close proximity, both geographically or chemically. And so in the cases in which we have very high sequence identity, or where we're actually seeing the MAGs compared to one another, are actually coming from either the same site but 2 different depths, or very nearby environments. So one thing that we've been asking is, okay, so what happens at a local scale? What goes on in microbial systems, and how does this variation actually look? And so to do this, my student Paula Rodriguez, working with also collaborators Mark Kleber and Jasmine Berg, sampled a specific lake in Switzerland called Lake Danio to collect a core and actually assess this genetic variation that encompasses death. And the reason we decided to pick Lake Danio is because it's a very interesting location in which you have sediments. So here we're not really looking at deep sediment, deep subsurface, but more shallow subsurface and actually sediments rather than hard rock. But it's the reason we wanted to go here is because here we have a lake that has undergone a transition from being completely ice-covered oxygenated lake into completely ice-covered lake into a mixed oxic lake, into now what today exists as completely anoxic water lake with an oxic surface. And so in this transition of the lake history over the last 10,000 years or so, you have, of course, sedimentation happening and burying various populations of microorganisms with time. And while this burial is happening, the microbial communities that are living there, it's not fossils. So I guess the main thing I want to say is we're not looking at fossil or dead material that has been stored in the sediment. We're looking at active microbial communities that are living and sustaining themselves deep underground. So we took a look at these regions in various ways, but what I'm going to talk about are sort of these genome-genome comparisons made from specific depths throughout the sediment core that represent various times in the lake's history. So what I want to see is who are these organisms that are able to survive and sustain themselves in the lake sediment, where the lake sediment that was deposited, the origin of lake sediment that was deposited has a different history over time, so different chemistry, and also has been subjected to burial at different depths in the scheme of the sampling. So when we look at what lives there, we just have this general picture. It looks more or less similar to what you might see as far as the amount of colors. This is more or less what you see when you look at a sediment profile of microbial communities, is often you have something rather diverse at the top on the shallow sediment, and as you get deeper, you start getting to a simpler community, right? That's becoming all dominated by this blue type. Now, the blue members frequently vary across ecosystem and environment, but the tendency to have something quite different in your shallow sediment versus something more stable looking and constant in the deeper sediment is looking here, is something pretty typical. And what we can do in looking at these genomes is we can look at these specific populations that are seen across these specific depths. And what we found were that there were 14 species found in 2 or more samples, and there were actually like 5 good representatives that were in like several of these depths. And so what it looks like when you compare on the local scale, we see that unlike the census of deep life that showed this very concentrated point of high genome identity, high genome alignment, we still maintain a high average nucleotide identity, which is expected because we're basically in the same location, but we're seeing a much greater tendency for more variation within the genome itself. And when we look at what's going on, for example, in this case of a specific species, which we find throughout all of the various depths, we can see that we have very specific gene clusters in the flexible genome, which are specific to organisms at various depths. And in particular, we can also see that in general, this trend is correlated to the type of sediment that's deposited in the layers, whether it's sort of pelagic, more pelagic or turbidite driven. And in particular, one of the things that is changing in the flexible genome are things associated with sensing to the outside. So these are things of transporters, transposases, and integrases that seem to be under selection in these environments. Of course, this Bathyarchaeota has quite a high variability over this short time scale in this population, which is quite nice, but we also have counterexamples, which are things like this, which is our planktomycetes, which looks more like that global picture where everything was stuck towards the upper corner. And a very interesting thing about this organism is, while the Bathyarchaeota is sort of a fermenter, we see that the planktomyces is this ammonia oxidizer. And it's actually it's very confined as well, while they've been being widespread throughout the sediment. This is an organism that is very specific to ammonia oxidation and only appears in the zone where ammonia oxidation is occurring itself. So this is some organism that is very much selected for its environment and is only really looking different, in particular through sort of viral elements, and in particular this one has a nice prophage that we find as a distinguishing feature, so a viral element inside of it that's distinguishing it from everything else. So 2 very different pictures of what species could look like, potentially tied to kind of what their role in the environment is. Something that is maybe evolved to be much more specialized, something that wants to be in a specific niche of ammonia oxidation, maybe has a tendency to keep more consistent genome architecture, whereas things like the Bathyarchaeota, even over some short timescale, short geographic distance, can have quite a high variety, something looking much more similar to what we saw in the Prevotella copri, changing many parts of its genome in response to its local environment. So this kind of leads to kind of a question is then what I posed in the beginning is, do these subsurface lineages evolve differently? I mean, in a way, I didn't get to showing this, but I'll just tell you that if you look at kind of the populations globally across the subsurface, you have a much lower what we call SNP frequency, so single nucleotide polymorphism. You have them much smaller than what we see in, say, a gut bacteria. These are less. We see lower amounts of divergence of the species, likely tied to the fact that the organisms living down there grow slowly. But, not really. This was sort of an open question, is do subsurface organisms evolve? What I think we can say, yes, we see evidence of drift and selection, and also evidence that there is activity like viral infection, horizontal gene transfer, etc., going on. And so I think to wrap up these ideas is kind of this question of what drives genetic changes and why is this complicated, why is this interesting, why we spend effort trying to understand is that actually, okay, these patterns exist, and there was this question, how does that actually happen? Because, there's two features of subsurface environments that I wanted to just mention here that are key to thinking about why they are unique, which is their population sizes are a bit smaller, which means your proximity to your neighbor is going to be much further away. And here's just some modeling by Jonas Tomka, which basically tells you about, okay, we saw a lot of viral signatures, but it's kind of a puzzle to us as to how it could happen because, if you look at the amount of cells, this is if you are at a cell density of 100 cells per mil or 10,000, which are more or less normal in the environment, and then you look at your virus to cell ratio, say 1 to 10 is maybe typical ranges. The amount of time that it takes for you to actually encounter a single virus could be on the order of days to several years. And of course, one has to also then amplify this question with the fact that viruses are very species specific. And so not every virus that you encounter will be able to infect you. So one big question is, why might all these viral elements and evidence of things like transposases and integrases be so predominant in these systems when they're low biomass? You may not have high encounter rates. And then the second question, this is sort of to do with those smaller amounts of genetic variation that we're viewing, is that actually as you go deeper, because you're not having this same amount of energy available, right? You're not having all this organic carbon, you're not having this oxygen, this turnover times of the chemoautotrophic systems are much, much slower. And so they can get onto the order of years, decades potentially, for the actual turnover of the community. So how are you actually getting fixation of these changes into your populations and actually showing these speciation over time scales? And so now I'll just pose my wild and one of my crazy ideas of why I think could be a cool way that could actually stimulate some of this process into why we actually get the diversity and see these things, is on this idea of a third water-rock reaction, which is when you break and grind up rock. And so much like radiolysis, when you break silicate rocks, you end up with these free-hanging radicals on the surface of your rock. And that radical is able to split water, and of course creates a lot of ROS, which is highly reactive, can create many byproducts, as well as can damage cells. And so this is actually something that we first started looking at in the context of some experiments we were running in what we call the Deep Life Observatory in the Bedretto lab, which is a very nice subsurface observatory that everyone has an open invitation if you ever want to come. We can all go on a field trip down underground. And so this is a 5-kilometer tunnel that goes in. It's in beautiful Ticino. So we're walking here, go straight into a mountain, and get up to 1.5 kilometers underground. And what we were doing was, through some different geothermal types of experiments, breaking and smashing rocks, and we saw this weird pattern that we wanted to investigate more. So we saw that, there seemed to be some changes in chemistry and also the biology that maybe has something to do with our process. And we found out, yes, that this reaction of breaking and splitting rock could be interesting. So we decided to look into it more. And this is the work of Andrew and our master's student, Andre, who's created many, many, many bottles of the water from this deep environment, with some with our crushed rock and some not. And what you can see is here, if you look at the treatments of what cells look like in the field that were bottled and then left to incubate over time, when you have crushed rock, we have this very sharp drop in our population, which also corresponds to an increase in viral production. So this is actually something quite interesting, is this large extinction of many lineages, then followed by a growth much later. So we have cell death due to presumably this radically mediated stress, because this is a very strong pulse of ROS into the environment. And we assume that this collapse, of course, releases many nutrients. We have the cells themselves as a necromass pool that can be consumed in some way, but also the crushed rock can also become more reactive and start leaching nutrients into it, in particular things like phosphate coming into the rock. And when we look at water chemistry as far as what's going on, this sort of increase afterwards seems to be related to likely sulfate reduction that's being triggered in these later stages because of a decrease in sulfate in the treatments. So a very interesting scheme that we've started looking at, and hopefully we'll have more on soon from some additional, now more genomics-based experiments that Andrew has been performing to actually see what these communities look like and what the changes in the genomes are. But I'll leave you with a thought about, okay, these mechanisms that can be quite sudden, quite explosive. You saw already that it can crash the microbial community, right? This was quite a dramatic drop in the population size. But we can also see that there's quite a lot of hydrogen produced very quickly. So this is 7 hours of a treatment of crushed rock interacting with water. And here I put it in nanomoles of hydrogen. And these are the numbers here. It's a bit small, but to match basically the estimate of those radiolysis models that I told you about before. So the amount of hydrogen produced per year by radiolysis through those various papers is on the order of, nanomoles of hydrogen per year. And here I have, by a rock, in a matter of 7 hours, I'm having this very strong pulse of hydrogen being produced in 7 hours. So of course, this is several orders of magnitude more production of hydrogen through simply a physical process that can happen. And this could be a very interesting potential, could have very interesting impacts on ecosystems and potentially the evolvability of subsurface systems themselves. So in a bit of a roundabout way, can get back to this, outlook and open questions of what we're thinking about is that, yes, there's a lot of diversity in the subsurface world. This diversity is not just an artifact of life's surface diversity being trapped underground. There's also actually processes that happen through the subsurface that seem to actually lead to selection and actually changing of flexible genomes of populations living in diverse subsurface environments. You can also see that water rock reactions can sustain complex microbial ecosystems, and that's by studying these patterns of diversity, we can actually see that specific things like specific traits of, say, something that is very specific to a metabolic niche, maybe is less amenable to, is less likely to potentially change its genetic repertoire, versus, say, some more generalists, which seem to actually evolve and change aspects of their genomes more rapidly. So this is, I think, a very interesting evolutionary question, is there actually, what is leading to organisms being, having a tendency to be more flexible or less flexible? What actually drives that pattern? So this is, I think, some exciting things that can be looked at and thought of in the context of microbial evolution. And, you know, just to leave some questions, because I heard some clinking outside, to the crowd, is a lot of talk is always about atmospheres, right? But when we talk about subsurface life, it's interesting because maybe we can get there and we can see and we can look for it. But can this deep life actually affect surface fluxes? Can this lead to some signature? And I'll just leave a thought there that actually there's been some recent work about how much CO2 fixation is actually done by subsurface life, and estimates say could rival the oligotrophic ocean. So rather high estimates, higher than had previously been expected estimates for CO2 fixation of subsurface life. And then lastly, I think everyone maybe has some thoughts potentially on, could we actually detect planets sustaining subsurface biosphere, which I guess is tied to fluxes, but some food for thought, and looking forward to more discussion. So with that, I'll thank my collaborators and happy to take any questions.